Welcome you all for the today's lecture. The lecture is on cyclohexane and substituted cyclohexanes and decalins. So in this lecture we will discuss about cyclohexanes, its substitution at different positions and their conformation, and about the decalins. So here if you see in more detail, so we will see here how does the substitution affects the stability with respect to check the conformation of the molecules, mono substitution and disubstitution. It means we have to see in substituted cyclohexane whether it is substituted from one at a particular site or whether it is having a two different substitution. Apart from that, in this overall, we will also discuss about the decalins, which is having two ring, and in this case, cis and trans forms we will also discuss, and we'll see how does these molecules within different conformation uh, and what type of arrangement is there. So we will discuss one by one. From here, once you will see the conformation of methyl cyclohexane. So this is the example of mono substitution. As we discussed in case of cyclohexane, the conformation you had seen over there, the cyclohexane is having different conformation, different forms. So in that individual forms, their stability is also very important, like you know boat form and chair forms, which is the two major forms. And if you remember in from our earlier lecture, so the different forms is having their different energy, so that decides the stability of the molecule. So here the more stable conformation of methyl cyclohexane is chair form. So now here the chair conformer. Why? Because we will see here, but with the example of taking the mono substituted molecules. So methyl cyclohexane has two possible chair conformation. First one is methyl group in an axial position and two methyl group in equatorial position. So what could be, and among these, which one will be the more stable and why? So here if you see this double arrow, half hook arrow, and this in equilibrium, so you can see this arrow represents the stability of the individual organic molecules, or here if you say in terms of the spatial conformation or the arrangement of the group on, as we discussed in earlier lecture, there is different position, axial, equatorial, axial, equatorial position. So in which position this particular substitution is there, that decides the stability of the cyclohexane. So here, as we said, here you can see there are different type of interaction will work. So if you put the numbering from the substituted group like 1, 2, 3, 4, so you can see from here also the numbering will go as the same fashion, 1, 2, 3. So you can see here at 3 position there is axial hydrogen. Also in case of cyclohexane in equatorial position there is also hydrogen atom. And here the substitution, if it is in substituted group is on the axial side, then you can see there is a interaction, and this is a steric interaction or steric effect. It means they will try to repel the molecule apart from each other. So it means this form will be lesser stable compared to the group when it is attached with the you know equatorial position. So here you can see in case of equatorial position, the all possible interaction, 1,3 interaction, which is generally called since at the atoms or groups, which is attached at the first carbon, and the interaction with respect to in a 3D space, if we see, so these are having steric effects. So here if you can see this another form which is equatorial, when the substitution is on equatorial position, so in that case the possible interaction is very minimum. So that's why this form will be more stable. So similarly, there are many more substitution you can check. So in case of monosubstituted cyclohexane, axial methyl group is more steric hindrance than the equatorial one. So here you can see how does equatorial and axial position is having different steric hindrance due to this equatorial methyl group is lying apart from each other. So there is no any kind of interaction like in case of 1,3 interaction in case of axial position or axial methyl group. So source of steric effect is close approach to axial hydrogen on same side of the ring at C3 carbon. So steric hindrance is called 1,3 diaxial repulsion. So due to this diaxial repulsion, this gives instability to the molecules. This gives instability to that particular conformer of the cyclohexane. So a chair interconversion occurs, but 95% of the molecules have their methyl group equatorial position. There could be a different interaction, interconversions occurs like chair interconversion, but the 95% of the molecules have their methyl group on equatorial position. So now you see like in monosubstituted, we have seen how the one group is getting to be interacted, like in case of methyl, it is getting to be interacted with their third hydrogen, which is in case of the axial position. So each carbon atom in cyclohexane has one axial and one equatorial position. So the dimethyl on C1, one of them is drawn axial and another on equatorial position. So each carbon in cyclohexane has one axial and one equatorial position. So the dimethyl on C1, one of them is drawn axial and another on equatorial position. The overall meaning what of this is, in case of disubstituted cyclohexane, the two methyl group will align. One will be on axial, because in this regular molecule it will not look which one is axial or equatorial. So you have to draw the molecule in their conformer in form of conformation, like chair conformation of the molecule, and then you have to put one methyl group on axial position and another on equatorial position. So this conformation is unstable due to 1,3 diaxial interaction. So we will see here how does in case of disubstituted or substituted as the example, how this is affecting the overall stability of the molecules. So we will see here, you can see from this molecule, once you see in case of the axial position of this methyl group with respect to, if you see the equatorial position, so equatorial position is having no such kind of any interaction. So like 1,3 interaction. So in that case 1, 2, 3 like this way, as you have to put the numbering so that you can see how does this, because this hydrogen is lying like this. If you see, just imagine the 3D structure of this like a flat structure. It is having like above the the plane, and this hydrogen this methyl group is also above the plane. So there is this interaction is there. So in this case, if you see, it can flip like this. There would be a flipping can happen, like as we discussed in earlier lecture. So there is a flipping will be there. So even though if you see, there is again these kind of interaction will be there. 1,3 interaction will be always there. So that's why the stability, as far as in terms of stability, so you can see from this sign, because this is having the equal chance of the conformer to exist, either this is in this form or it can flip to another conformer. So no matter in which conformer you are drawing, and you can watch it like this way, how does these interaction unstabilize the overall conformer in case of cyclohexane, in case of substituted cyclohexane. So now, like cis trans, there is isomerism. You also know what is the isomerism, and conformational structure of cyclohexane. Once we see here, so cis trans 1,4 dimethyl cyclohexane. Uh, so we can see how does these are seen. So the trans means the two bulky group, whatever the bulky group, like here in case of dimethyl cyclohexane to methyl group is comparatively more bulky with respect to the hydrogen atom in opposite side. So if these are on opposite side, then it will be said as a trans. If these are like here, one is in axial position, another in equatorial position. So if you know to draw it like this is axial as we discussed in earlier lecture. So if you remember that, you can easily find out which one is having axial position and where the group we have to put, so that it can convert into in their different forms, or cis or trans form. So cis means the two bulky groups in the same site. How do we know two groups are cis or trans? So here this example can help you understand better way. So generally, to recognize trans disubstituted cyclohexane is, notice that one group is attached by upper bond of the two to its carbon, and one by the lower bond. So here if you see in case of cis, so this is like cis forms. If you see the cis form, and because these are anti to each other, however these are the axial position with respect to cis. So you can say 1 2 3 4, 1 4 axial equatorial will be the cis form. Okay, since this is the staggered, you can say with respect to these are just anti to each other. So this methyl group and this methyl group is having, this is called upper bond, and this is upper bond, or you can say in case of axial or equatorial terms, you can say 1,4 axial equatorial will be cis. If the group, if the substituted group is on axial or equatorial position, then that will be cis form of that particular substituted cyclohexane. So 1,4 dimethyl cyclohexane. So this is cis 1,4. Groups are cis if both are connected by the upper bonds, or if both are connected by lower bond. So if it is having lower bonds, then the same lower bond will be also known as, or you can say axial equatorial or equatorial axial. If suppose this is equatorial, then this will be the axial position. So, or you can say in terms of lower or upper, so both will be, in that case it will be lower. So here if you say in case of trans, so one will be at upper bond, or you can say with respect to this carbon, this is equatorial position, so equatorial position, and this will be the equatorial position. So if the atoms are this is equatorial and this is having equatorial, so in case of upper bond or lower bond, so this is the lower bond, this is the upper bond. So upper bond and lower bond, if the atoms or groups are attached, then that will be called as this trans form of that particular substituted cyclohexane. So similarly, you can see here, like in case of trans, you can see here, in case of trans, here axial position, both are trans, but here if you see equatorial, or you can say one is above and another one is below, down. So the trans 1,4 cyclohexane 2 conformer, trans diaxial and trans diequatorial. So both trans 1,4 has 2 conformer, if you are putting them both are axial or both you are putting like equatorial position. So according to that, we can have also put them in a different form. So now in case of cis conformer like cis 1,4-dimethyl cyclohexane, we have to see how this conformer in case of chair conformer, here you can say the cis conformation of this equatorial axial or axial equatorial, and in case of 1,4 di substitution and axial equatorial, here this group once it is going to be flipped chair, this is ring flipping over here. So this is moving towards the axial position, this methyl group, and this comes to equatorial position. So in which one group is axial and other is equatorial in both the of the possible conformation of the chair, either it is a ring flipping. So that is called cis conformer of 1,4 disubstituted cyclohexane. So this substitution could be any kind of substitution. In place of this group, you can change with many more other possible groups, but their isomerism or their cis and trans conformation will be according to, you have to see if suppose one group is at axial position and another is equatorial position. So accordingly you have to designate them cis conformer, and as we discussed earlier like slide, so similarly you have to designate them trans conformer. So now here the another which is very important part of the molecule in case of to see their stereo and to see their conformation, to see their structure. So another is decalin, which is two cyclohexane ring fused ring, and it is having the name bicyclo 440 decalin. So this decalin molecules, if you see in their different conformations, so saturated analog of naphthalene and can be prepared from it by hydrogenation in a fused state in the presence of catalyst. So this can be prepared with the help of hydrogenation reaction. If you see how to prepare it, so it is decahydronaphthalene basically. uh, you know it is once you can take the naphthalene molecules, and once you will hydrogenate it, then you will see this is tetraline, this another molecules will form out. So further once you reduce it with hydrogen, then you will see there is two forms, cis and trans form of the decalin will be, you will get the cis and trans form of the decalins. So this is cis trans form, and another one is cis form. So here you can see the in which direction the particular hydrogen is attached with that fuse along the fuse ring. So Sase and Mohr proposed Decalin is a puckered stain-free two-ring cyclohexane existed in two forms that could interconvert only by breaking and reforming bonds. In 1925, Walter and Huckel isolated the two postulated isomer of Decalin. So here this is what all about the conformation of the cyclohexane. Here they had Mohr proposed the puckered stain-free two rings of cyclohexane existed in two forms that could be interconverted only by breaking and reforming the bonds. So if you see, decalin is a hydrocarbon which has six-membered ring like those of cyclohexane are expected to be most stable in the chair form. So here again you had seen in case of like cyclohexane chair conformer is the most stable form. So similarly here the chair is expected more stable form. However, there are two possible ways in which two chair conformation of cyclohexane can be joined. So like two chair form, how can they can join to each other? So there are like this form which is the cis decalin, and when they are lying apart from each other, the two rings if you see then this is called trans decalins. And if you show in structural point of view in regular structure, so here you can see the hydrogen, the arrangement of the hydrogen towards the direction of the viewer at the fused carbon. These two carbons are fused to, this is the common carbon for both the rings. So here this hydrogen is rear side of the cyclic ring, these two rings. So these forms are very important with respect to synthesis, because when we synthesize various natural products, so during the course of synthesis of natural products, these decalins makes the substrate or makes the building blocks of those higher like steroids and all naturally important materials, which is already in the nature and can be also synthesized the similar analog in the laboratory as well. So here trans-decalin, if you see here, it is conformationally logged framework. It no undergo any ring inversion, having if you see the chirality, in terms of chirality, a chiral, you can have a plane, you can pass a plane over there, equal halves, you can. So center of symmetry, midpoint of the 9 and 10 bonds. So here you can say center of symmetry is there, and C2 axis passing between C2, C3, C9 and C10 and C16, C17. So all axis of symmetry is present here. So if you remember when we discussed previous lectures about the different kind of symmetry elements. So if there are any kind of symmetry elements, then obviously you cannot say whether that will be chiral or not. So to check the chirality, to check their stereochemistry, you have to see the symmetry elements, symmetry properties, which is very important to check the molecules in case of like such kind of ring, such kind of cyclohexane rings or fused cyclohexane ring, which is the example of decalin here. So you need to know. So if you see the cis decalin, so cis decalin, if you put it into like a Newman projection, so it looks like this. You can see here the different carbon and the fused carbon, and they are having here. So we can see the symmetry elements with respect to this by putting the molecule in the Newman projection formula. So there is C2 axis of symmetry is there. So is C2 isomer is carbon is isomer is without chiral 2. In case of cis decalin, there is even some kind of symmetry element is there, but without chiral center. Two-fold rotation symmetry axis going through the center of 1,6 bond, but no reflective symmetry. However, the chirality is cancelled through a rapid chair flipping process, because sometimes it is having some particular kind of chirality is there, but due to the ring flipping the molecule into its mirror image, hence that the compound is not resolvable. So because due to their rapid flipping, so it is very difficult to to segregate them. Has a folded structure with convex and concave sides, so that the steric reflection are unequally distributed on the two sides. So by this way, by putting in a Newman projection, you can have a different arrangement of the atoms. You can check with the particular type of cis decalin here if we are talking about. So cis form can be converted into in their different conformation. So these are the conformation. You have to just see how we can project them in a projection formula, which we have already discussed about the projections of the molecules, how the projection formula like Newman, Sahos, Fisher projection. So here Newman projection is quite important when we are discussing this. So different carbons, and there is different kind of symmetry elements, which is also very important to understand. the Decalin and their cis transform and their chirality, their nature of either they are chiral, achiral, and all. So this helps to explain the molecular structure properly, and finally these kind of stereochemical analysis helps to assess the course of reaction, whether the reactions in a synthesis, especially when it comes with a very higher molecular synthesis like steroids and higher alkaloids. So these decalins helps to take as a substrate and helps to propagate the reaction in a certain manner. So this is what is looking. So if you see the decalin we have here, if you say, as I said, these are a fundamental framework of steroid, uh, is a tricyclic carbon framework, because during the synthesis these higher steroids, which is having a very complex molecules and takes a very long steps to synthesize in lab. So these are the fundamental framework as well, and you can take the starting from these kind of substrate, and then you can frame out the special kind of steroids in the lab. And here if you see the trans-decalin style fused cyclohexane rings are the basis for steroids. So these are taken as a base or starting for to synthesize the steroid molecule. Many have different groups, like especially once we are talking here about methyl groups at C10 and C13 position. So these type of insertion of the individual groups on the particular carbon atom, that helps also to synthesize the new type of molecules in the product chemistry. So in the field of natural product chemistry, where you studied about alkaloids, terpenoids, and many more plants, hormones, and the various other constituent of the natural products. So during those are being since extracted from the nature and can be uh, in case of also this will be very important decalin structure in retrosynthesis, because that is just the opposite to what you generally do with the synthesis process. So this is just the opposite of retrosynthesis, is the opposite of uh, organic synthesis, you can say, because normal synthesis you start from the substrate and you makes a certain certain product. But in case of retrosynthesis, you have to start, you have a molecules, you have the product, and you have to see which type of simpler the molecules or simpler atoms that particular kind of steroid has been synthesized. So this is the reverse synthesis, means you have to find out what type of the starting material we should take so that we can approach to synthesize the product. Like this happens in case of the natural products chemistry as well. Since if suppose you extracted certain certain steroids or certain natural products from the plants or from any animals and any kind of particular chemicals. So if you confirm or if you characterize that molecular structure, so if it is a very complex structure, then the challenge is how we can synthesize that molecule in a laboratory. So in lab, you have to see what could be the possible starting material, what could be the possible starting substance which can make the framework of that particular kind of steroids or particular kind of organic molecule. So overall discussion, if you see, we had have a discussion on substituted, cyclohexane, their different conformation. We also discuss about the mono substituted, substituted conformation, and how does they are interacting to each other, and what type of interaction is there, which form will be more stable, and what is their, the conformation. And decalin we also see seen their cis transform and their fundamentals, how does cis and transforms can be put into different projections so that we can find out. which form will be useful when we talk about to synthesize a special kind of molecules, like as in case of steroid synthesis. This is very important starting material for the framework of the steroid molecules. So here some of the references, different reference materials from where these materials has been taken for the lecture. So different websites, online resources, and these materials are only for the instructional purpose and not for any commercial use. So here you can say various books are there, so you can refer those books. So I thanks you all for watching this video and listening this video lecture. Thank you so much.